Good morning, everyone. As Matt has mentioned, I am Lutholwethu Mabaleka. Today I'll be sharing the findings of a chapter of my master's research project that is looking at fish habitat associations in the Marine Bay region of the Knysna Estuary. So estuarine ecosystems are widely known to be highly productive and that they serve an important nursery function for juvenile marine fish species. So within those systems, there is a wide range of habitats that juvenile fish use as nursery areas. However, it is important to note that juvenile habitats are only referred to as nursery areas if they contribute higher recruits to adult populations. Literature on fine-scale habitat complexity have highlighted a positive correlation between structural complexity and juvenile fish abundance and diversity. And with structurally complex vegetated habitats linked to high food availability and structural refuge provided to fish located in those areas and making them high-quality nursery habitats. So the area of interest, which is the Knysna Estuary, this estuary is the largest estuary in the Southern Cape coastline, and it has gained recognition as one with the highest conservation importance in South Africa. It also hosts a wide range of habitats, some with an unknown nursery function, because most studies on estuarine habitats and nursery function in the Knysna Estuary have focused on seagrass and salt marsh. So despite the ecological importance of estuarine ecosystems, they are one of the most threatened systems globally. And an example, the critical habitats within the estuaries are threatened by development, bait collection, and nutrient pollution. And in addition to, among other factors, and in addition to that, there's also an occurrence of invasive species such as the red-algae Asparagopsis taxiformis that is now widely distributed in the Knysna Estuary, and it occurs within the endemic seagrass Zostera capensis beds. So a recent study in the Knysna Estuary looked at sub-adult and adult populations in the Knysna Estuary, throughout the Knysna Estuary, to find priority areas, and it found that the Marine Bay and the lagoon regions are hotspots for juvenile sparids that are caught in the shore-based line fishery. However, the fine-scale habitat-specific early recruitment fish assemblages associated with habitats within those regions are unknown, and also the nursery function of the widely distributed Asparagopsis taxiformis is unknown. Therefore, this study aims to evaluate habitat-specific fish assemblages in the Marine Bay and lagoon regions of the Knysna Estuary. And to achieve this aim, we have those two objectives, which is to determine the influence of habitat features on the early juvenile, on nursery habitat selection of the early juvenile marine fish species. And the second objective is to determine fish assemblages of the early juvenile marine fish that occur in the different habitats. So for this study, we collected data seasonally using a small mesh seine net. We also recorded physicochemical parameters, and also we took plant samples to measure the different structural complexity indices in our seagrass sites. So to answer objective 1, we created a spatial map on ArcGIS to show how the different sites that were surveyed varied in structural complexity. And we also, to determine the influence of habitat features on the early juvenile fish, we performed a Spearman's rank correlation coefficient on RStudio. So for objective 2, we used we used multivariate analysis to determine the distinct fish assemblies across the habitats on Primer, and also our univariate analysis was conducted on Statistica. So this is, we had 3 sampling periods in spring, summer, and winter, and this is how the different sites that were surveyed varied in structural complexity. Most of our sites were located within the widely distributed seagrass in the Knysna Estuary, and some were sand, and we also had Asparagopsis sites. And it's important to highlight that looking at the winter spatial map, we can see that there is a presence of we had an algal bloom in winter, and like literally most of our sites were smoothed in alva, and the sites that were previously long seagrass were now short seagrass, and we're assuming that it's the effect of the algal bloom. So this is how the seagrass sites looked like, smoothed in alva. So our results showed that at fish community level, there was overlapping in our sites, with A and B consistent in statistically as well, that they did not show any significant difference in our fish community. However, for season, which is like plot C, it did even though the sites are still overlapping, but statistically they showed that there are distinct fish communities based on season. So for figure 7 shows how our total fish, our relative abundance varied among our sites sampled, and A shows the relative abundance and B shows species diversity among our habitats. And figure A, it shows that Asparagopsis had the highest catches of our early juvenile marine fish, followed by long Zostera capensis mix, and this was consistent with our statistics as well, because there was a significant difference among the habitats that were surveyed, and particularly in Asparagopsis and sand, and also Asparagopsis and short Zostera capensis. And then looking at B, which is the species diversity, was structurally complex vegetated habitats. They showed the highest species diversity compared to the sand, and consistent with this this visual presentation, statistically also one-way ANOVA reveals that indeed there was a distinct significance in the species diversity among the habitats that were surveyed, particularly in the long Zostera and the sand habitat. So at species level, looking at the far left, we have blacktail there, which is Diplodocus capensis. It showed association with all our habitats that were surveyed. However, the highest catches were in the macroalgae Asparagopsis taxiformis. Similarly, the marine estuarine-dependent Rhabdosargus holubi also showed association with all the habitats that were surveyed, but most catches were in the long Zostera capensis. And then our strapy, which is the Sarpa, showed it's herbivorous, and it showed association with seagrass and Asparagopsis, which is the macroalgae. Okay, so in conclusion, at fish community level, we found that the early juvenile marine fish species do have differences, are significantly different among habitats that were surveyed, with the highest catches in the red algae, and structurally complex vegetated habitats. They showed the highest species diversity compared to simpler habitats. And at species level, despite the occurrence in all habitats surveyed for some species, some of the dominant early juvenile marine fish species, which are also recognized as important fishery species, showed strong association for vegetated habitats. And in terms of conservation spatial planning, these findings highlight the importance of conserving a mosaic of habitat types within estuarine protected areas, as different habitats support distinct components of the juvenile fish caught in the recreational fishery. Thank you very much.